So our next speaker is Rafael Couñago from the Structural Genomics Consortium and the University of Campinas, who will be telling us about the development of novel tools to guide the discovery of new cell-permeable on-target anti-infective compounds. So,
thank you so much, Natalie. I'm just going to share my screen, so please let me know if you see it. It should be coming up.
not yet. No.
Not yet, okay. Let's see if that's the trick.
Yeah, it's getting there.
All right, excellent.
Okay.
Yeah, perfect.
All right, so thanks for the invite. It's a pleasure and it's really a privilege and an honor to be talking to this audience today. And I just wanted to celebrate a little bit more of the Protein Data Bank. 50 years is not that we can do to celebrate it enough. Of course, there's all this hype about the modeling and the new capabilities that was only possible because PDB is freely available for everybody, right? But there's another side to it that many people are aware, but I just wanted to highlight that if you are from a developing world country where in the 90s it wasn't that easy to get your hands on journals, sometimes you could get actually the coordinates before you had access to the journal on your library. So this meant that many lives were touched by this initiative. And so many thanks. It changed my life, and I bet it changed many other people's lives as well. So with that in mind, right, with this explosion of structural biology, you know, Brazil is one of the countries in South America that invest heavily in structural biology. So it's a privilege to be talking to you today, especially from a country where we have so many excellent X-ray crystallographers. So our lab, it's located at the University of Campinas, and it started as a structure-based compound development center. So our idea was to make chemical probes that I'll talk a little bit more about it in a second. And it's truly a success story in Brazil for a public-private partnership. So today, we have about 18 postdoctoral researchers in our group, and the fundings for our group comes from a diverse source of partners, many of them from the private sector. So it's really a privilege to be working in such an environment. Just to let you know where we are exactly, we are in Campinas, which is a city of about 1 million people, and about 90 clicks from São Paulo, that's about 55 miles, and about 400 kilometers or 250 miles to Rio de Janeiro. And we are only a small walk away, a short walk away from the new synchrotron in Brazil, Sirius, which is a fantastic achievement for Brazilian science and arguably one of the most ambitious scientific projects in the whole South America. So it's pretty a privilege to be so close to this amazing facility. And this is our building. We occupy this first floor here, and that's where all the science happens. So my group at the Center for Medicinal Chemistry at UNICAMP, it's concerned with developing chemical probes. And our thinking is that chemical probes are a good tool to advance new protein function and try to find new therapeutics or new therapeutic targets, better. Chemical probes have a big advantage over other tools when we are trying to investigate protein function, as the action of these compounds are time and concentration dependent and require no genetic manipulation of the biological model under investigation. In our lab, structural biology, of course, plays a big role in understanding compound binding mode and allow us to develop more potent and selective ligands. But more recently, we have also been using structural biology to try and develop new assays to see on-target effects of these compounds in cells. Our lab started in 2015 as part of the Structural Genomics Consortium Chemical Probe Program for protein kinases. Of course, kinases are unquestionably one of the most important drug targets at the moment. And but it's important to realize that only a small percentage of all the kinases that the human genome can encode are actually understood. Their functions or their mechanisms are actually understood. So the one way that we thought we could advance new therapeutic targets was by creating small molecule ligands to all of the kinases, or to all the kinases that didn't have a binder. And I'm just going to share a little bit with you the story of our ChemKK2 project, which is the calcium comodulin dependent protein kinase, kinase 2. It's an understudied kinase in the sense that it doesn't have a chemical probe that goes with it, right? Of course, ChemKK2 is the topic of many hundreds of papers, but we felt that the quality of the ligands for this target wasn't condescending with its relevance in the organism. So a little bit about the protein, it's a kinase because it has a kinase domain, of course, but it also has other regulatory bits attached to it in the N-terminal and the C-terminal. And one that I want to tell you about a little bit is this autoinhibitory region, AID. And the AID is a region that binds to calcium when it's bound to comodulin, and that frees the enzyme to do its function. So it becomes catalytic active once this little bit of the protein binds to calcium comodulin. And this was done by NMR, this structure back in 1999. But more recently, using a protein exchange coupled to mass spec, it was shown that this little bit here actually, which is part of the autoinhibitory domain, it can interact with the binding site, the ATP binding site of the kinase when in the absence of calcium comodulin. So that kind of gives an explanation for why the protein is inactive in the absence of calcium comodulin, and when this partner is available, then the protein is active. And I like to show this structure because it's one of our structures that we solved and we deposited as soon as we could in the PDB back in 2017, right? And this Czech group that did this work published this in 2018, but our structure was only published again in 2021. So if we had waited to put this structure out for the publication, then they couldn't have used our structure, which is really great. And this structure is the only structure at the PDB of ChemKK2 that shows this little bit of the protein ordered in the crystal. Most of the other examples we have of ChemKK2, this is a very flexible region, so this is disordered. And ChemKK2, of course, it responds to calcium concentrations inside the cells, and that, of course, is a major signaling molecule that controls many pathways. So this is upstream of many more famous kinases like MK, and controls a series of cell states in the cellular metabolism, and also it's involved in many diseases. The problem is the link between ChemKK2 and its function and its disease involvement in many instances is based on the use of this small molecule. So in about 200 publications, you have the use of this STO609. And it's not a very selective kinase inhibitor, and also it's not very potent in cells. So here is a Western showing the phosphorylation of MK, which is one of the substrates of ChemKK2. And we can see that there's a little bit of reducing of this phosphorylation, but it's still persistent. So all these conclusions, maybe they were obtained not using the best chemical tool available. And when we started this project, which really spearheaded by David Drury at SGC UNC in Chapel Hill, there was no potent and selective compound for ChemKK2. So he started from a promiscuous but potent skeleton from GSK program. And throughout these 5 or 6 publications we have now out about this project, we solved many crystal structures and developed many biochemical assays, and to get to what we now have a chemical probe for ChemKK2, where it's very selective and very potent and with a very good biological function or activity on the substrate. And it's really up to, or it's really merit of David Drury to look at the structures, all the ones that we solved, about a dozen or so, and look at the backbone of the scaffold of the molecule to try and make it simpler. So what he did was to open the ring, but keep the hydrogen bond to the hinge region of the kinase, and that increased the specificity many fold. But as we were developing this ChemKK2 chemical probe program, we realized that this showing target engagement in cells was a critical part of our research program. Because biochemical potency is only half of the story, or even less than that, right? Your chemical probe must be cell permeable, it has to hit the target, it must be bioactive. And asking a collaborator to run hundreds of Westerns for dose-dependent curves, to generate dose-dependent curves for analogs of your chemical probe, it's really something, it's an ask. It's too much to ask of them, right? So, of course, we try to screen it at a fixed concentration, but then you don't really have the potency of that molecule. And right from the beginning, we were paying attention what Promega was doing on this, they call it nanobret. It's a Brett-based assay where you fuse your target to a luciferase, and you fuse your ligand to a fluorophore that's compatible with the luciferase. And if everything goes well, this Brett probe, as we call it, goes into the cells, binds to the target. The energy from the luminescence from nanolook gets transferred to your fluorophore, and that emits fluorescence at a different wavelength. If you have a ligand, it disrupts this interaction, and now your Brett signal goes down. So you have a real-time manner to assess the biochemical potency of your small molecule ligand within a living cell. And we thought, this is great, let's do it for every single chemical probe project that we have. Unfortunately, not every single commercially available reagent that Promega sells is compatible with the target that we wanted to study. So this is where understanding the structure of ChemKK2 really helped us, because if you remember, the autoinhibitory domain goes back into the active site and prevents ATP binding. And all these probes, they are based on ATP. So what we did to make this work for ChemKK2 was to remove all the regulatory regions and only have the kinase domain fused to nanolook. And now it worked pretty well. So this is a dose response curve for some of the analogs during our chemical probe project. And you can see that the probe candidate number 62 is really potent in cells, way more potent than many of the other compounds that were really good biochemically. And this got us really excited, because that meant that if we know the structure and we know ligands, we can tweak different systems to come up with a target engagement assay in cells. So we took this and we decided to go into leishmania kinases. Of course, we are a lab that worked for many years on kinases, so we had that knowledge base. And also, being in Brazil, we are very sensitive to the neglected tropical diseases arena. And also from collaborations with Jeremy Moultrie at the University of York, we knew that many of the kinases in leishmania, about a quarter of them, are essential for the parasite to infect the mammalian host. So we thought this is a really good system to try and get this BRET assay to work. Of course, leishmania is very challenging. That's why there are not that many good drugs around. They live inside a vacuole, acidic vacuole inside a macrophage. So for a compound to be active against leishmania, it has to cross multiple membranes and different environments in terms of pH and remain active to hit its target within the parasite. So this is the schematics of how this BRET assay should work in leishmania. You have your probe, you have your target fused to nanolook. And when you add a ligand, displaces the probe and reduces the BRET. And from Jeremy's work, we knew CLK1 was an essential kinase in leishmania. And also from his work with Novartis, we knew that there was a ligand for the T-brucine, the trypanosoma brucei enzyme, or CLK1. So we thought, okay, we have a target, we have a ligand, let's see if we can make this work. Making the recombinant leishmania wasn't that hard, so we got leishmania to express CLK1 fused to nanolook using CRISPR, but none of the Promega reagents that were available for the human CLK1s, they worked for leishmania cells. They worked in vitro, okay, with purified protein, but not in cells growing in culture. Only when you actually electroporated the leishmania, which wasn't really what we wanted to do. Again, we went to the PDB to look at the structure of the T-brucei CLK1 bound to this Novartis compound, AB1. Unfortunately, the compound was a covalent inhibitor, which is not ideal to be the scaffold for a BRET probe for a competition-based assay. So we had to identify novel ligands for CLK1, which we did using thermal shift assay. And to our despair, we noticed that many of the fluorophores we tried didn't really work. So we attached different fluorophores using different linkers with different lengths to the ligand, and many of them just failed to generate BRET when you added it to media containing leishmania that was expressing the nanolook fused CLK1. But one of them did. So, okay, we only need one. So we moved ahead and using a non-fluorescent version of that BRET probe, we showed that we could displace the BRET probe from the target within cells, and also with a different ligand that's unrelated to the probe that came from the DSF screen. So we were pretty happy with this, with the system, and we felt confident that we could go ahead now and try to find a ligand that was cell-permeable and could displace the BRET probe from leishmania within living intact cells in culture. And we compared the result with our DSF screen using purified proteins. So satisfyingly, we did find compounds that were very potent in vitro with the purified enzyme, but just couldn't displace the probe in cells, right? This was kind of our whole idea, that this assay should really help us accelerate the discovery of new compounds, because it's going to be in cells where the permeability problem is, well, it's going to be a problem, but we are going to overcome it. And also we found, like this one by the highlighted by the purple arrow, that was quite potent both in the biochemical assay and the cell-based assay. And we confirmed these hits using IC50s or enzyme inhibition assays to calculate IC50s for the compounds. And this orange, the one that's highlighted by the orange arrow, for example, has a 30 nanomolar potency with purified protein. But when we tried to kill leishmania with that, we just couldn't. So again, that was pretty good finding that confirmed our initial finding from the high throughput screen. And conversely, the one that highlighted by the purple arrow that has an IC50 of 200 nanomolar, it did kill leishmania with 2 micromolar, and that's with intracellular mastigotes. So this MIC is not that bad. 2 micromolar is actually pretty good for leishmania. And even more satisfyingly, if you overexpress the target in cells, then the leishmania lost sensitivity to the compound about threefold. So when we overexpress is actually 17 micromolar or so. I forgot to put the number here. So this again was a confirmation that our assay identified a compound that was effective against host cell leishmania, and its activity was because it was hitting the target, right? So I think at this point, we were pretty happy with the assay and the way we developed it. And we thought, okay, so this is really good. So what's the other system we can try to help and accelerate drug discovery for? And
just a minute or two, Rafael.
Okay, thank you so much, Natalie. And we thought, okay, let's just go and move it into bacteria. So we first tried with E. coli, which is a gram-negative. So even though it's a domesticated E. coli from the lab, it's an actual gram-negative, right, with a very sturdy cell envelope, efflux pumps, and so on and so forth. And it works in a similar way as I showed you for leishmania. And again, we went to the PDB to get the structure of DHFR, which was our target for this, and bound to trimethoprim, which is an antibiotic that's known to hit DHFR. And we found a position where to put the fluorophore attached to it. And we show that you can do that. So if you do a titration of your breath probe into living cells, those are happy E. coli cells growing culture, your tracer goes in and interacts with the target that's fused to nanolook. And if you just use dark trimethoprim, the one without the fluorophore, it displaces the breath probe. So the whole system also works in E. coli. But as opposed to when we did with leishmania, the hits that we found that displaced the probe from DHFR, many of them didn't kill the bacteria. And that was puzzling for a while. Why is it that now our MICs are not following what we see with the breath assay? And the answer for that was because the efflux pumps were actually activated or actually having an active role in getting those compounds out. So we could only get decent-ish MICs with our compounds once we started deleting the efflux pumps. And we did that by deleting Tol-C, which is a component of many of the efflux pumps in gram-negatives. And the other really cool experiment we did with this assay was this compound residence time on target within the cells. So this works a bit differently from the target, from the tracer displacement assays, in the sense that you first incubate your cells with a compound, and then you add your probe. So when you do that, and if you dilute the antibiotic before you add your probe, then you should see the recovery of breath over time. And you see that when you use the MSO. right, for both wild type and tall C mutants. But if you have compounds like compound A, it's pretty good. It hits the target and stays there. You dilute the antibiotic, add your tracer, and you don't recover much of the breath signal. But for compound B, that's not the situation. You incubate the cells with compound B, wash it off, add your tracer, you only recover a little bit of your breath signal. But if you have the deletion of efflux pumps, then you recover a lot of that signal. So that means that the tall C is actually getting rid of that compound. And if you find a way to block efflux pumps, then maybe that compound could be a good candidate for an antibiotic. So this is what I wanted to share with you today, and I want to thank you again for the opportunity. I couldn't have done this with all the collaborators that we have. David Drury at UNC and Kara Wells that was working at UNC at the time. Now she's at GSK. John Scott in Melbourne. Carol did most of the work with leishmania, and Priscilla did the biochemistry for the leishmania work. Stanley did the tracers and the chemistry for the project. Matt Rovers, it's a great scientist from Promega and really helped us get this out of the ground. Juliana and Jeremy at York also were huge support to do the genetics on leishmania. For bacteria, this was a fantastic work from Rebecca, a master's student in the lab that just graduated, and Kyle helped us also with some structures. And I couldn't have done this without the support of SGC, of course, Promega, and the funding agencies in Brazil, and the beam lines also that we got the structures from. Thank you very much.
Thank you, Rafael, for this wonderful talk and example of forward thinking of how we can assess these therapeutics in the cellular context where it's so important. It's just gorgeous. We had time for maybe one question. Zaule Ma says, thanks for bringing up the work on CLK1 from Novartis. Did you assess the specificity of non-covalent CLK1 inhibitors against other kinases in T. brucei and human kinases such as human CLKs?
So we did try to make a non-covalent version of AB1, but it wasn't very potent. And this is reported for T. brucei as well. One thing is the Novartis compound, I believe it was developed for the mutant form of the human enzyme. So it doesn't really get into the wild type human CLK1s. And so I think this is also a good strategy to look at. You don't want to be working with compounds that you know are going to hit the human kinase eventually. So I think that was pretty smart from Novartis to look at the compounds that they were interested for mutant versions of kinases, for example.
Thank you. I think in the interest of time, we're going to move forward, but I'm sure that Rafael would be happy to answer further questions in the chat. So please join me in thanking Rafael for this excellent talk.